Well, welcome to Wave Club, everybody. I'm happy that you all are here. I hope everyone had a good holiday. Here in the States, we had a holiday yesterday, and it's the first day of campus of students on campus here. So for me, this is a fun beginning to the fall. And thank you all for your patience with our new Zoom link and a couple of emails you received. I think we have things sorted out, and it should be a little bit easier for everybody to access Wave Club in the future. So I'm happy about that. And Irfan did a lot of really great work getting organized for that. This is Wave Club. As you know, this is an online meeting group dedicated to traveling waves across species in the brain. As long as it has a brain, it has waves, and that's part. And well, maybe one day we'll do Wave Club with non-brain organisms. We shall see. Our speaker today is Edward Ester from the University of Nevada at Reno. Here is a obligatory photograph of Edward outside and exploring nature. Edward is going to talk about traveling waves in humans. The title of his talk is Traveling Waves Link Human Visual and Frontal Cortex During Working Memory and Guided Behavior. That's today. So Edward, thank you very much for coming. And the plan for today, is that Edward's going to present for about 40 minutes, and then we have some time for questions afterwards. Edward kindly said that he's happy to take small clarification questions in the middle. But primarily, the best way to what we really like to do is have a robust question and answer session at the end where we can really be interactive. And because traveling waves are such a cutting edge thing, I think it's useful to talk about them. So what we would like to do is anybody who has questions to type them into the Q&A feature at the end. And then what we'll do is that Uma is going to curate the questions and answers and bring people on stage virtually to ask their question to our speaker, and we can have a little bit of a discussion and a back and forth. And so if you put your questions in the Q&A feature, we can be organized and do that. And just before we get started, I want to mention our Wave Club YouTube channel here, where you can see a recording of all the past and future Wave Club events going forward. Did we get Edward's permission to record?
Yep.
Okay.
After that.
All right. So we'll be recording. We plan on recording Edward's talk as well as all the previous ones, unless Edward has a problem with it.
No, go for it, please.
All right. Great. I think that's it. So I'm going to turn over the stage to Dr. Ester. Bard, Uma, Irfan, did I miss anything? Okay. All right. Take it away. Thank you, everyone, for coming.
All right. Can everybody see that?
Looks great. And your campus looks very nice, I have to say.
Thanks. No, it's good. So we are kind of a hidden gem in that we are nestled right on the eastern edge of Sierra. So when I'm not homesick, I am over at Lake Tahoe every weekend, which is quite nice. Thank you to Josh and Bard and Uma and Irfan for organizing this. And also, I know that Uma and Irfan have been dealing with a lot of technical challenges recently. So a round of applause to both of them for getting all this put together and for kind of working out the kinks. So I'm going to talk today about, this is really a brand new direction for my lab, this traveling wave phenomenon. And really some of this work was inspired by some of the work that Josh and Bard have done in the past, as well as some previous EEG work that's come out the past couple of years. So Laura Duguay, who's presented in an earlier version of this meeting, was kind of inspirational for a lot of this stuff as well. So let me start off by first acknowledging that I'm really not the one who should be giving this presentation. A lot of the work that I'll be talking about today was done by a former postdoctoral fellow in the lab, Dr. Kanwang Lo, who happily has since moved back to his native China and is setting up his own lab at the University of Shenzhen. However, it's about 1 o'clock in the morning his time right now, and so he asked me to dive on this grenade for him, and I'm more than happy to do so. But really, a lot of the credit goes to Ken Wang on this, and so he and I are kind of continuing a collaboration on this work going forward. So let me start off by talking a little bit about what we do in our lab. So this is a graphic that you might find in any kind of introductory cognitive psychology or cognitive neuroscience textbook, talking about the relationship between kind of three critical cognitive functions. So on the early end of things, we have sensation and perception. So there's interpretation of what we perceive and what we sense in the world around us. A lot of that happens in a limited capacity system that we call working memory. And then ultimately, we need to act on the information that appears in our environment around us. And so a lot of people have been studying perception and working memory and action for many years. We know that there's bidirectional links between many of these different processes. And one of the things that we're really interested in my lab is this link between working memory and action. You can even, in some sense, conceptualize working memory, although it contains information about what happened in the recent past. It's really about using that information to make decisions and to perform actions in the present or in the immediate future. So you can actually conceptualize as working memory as being an action-oriented system. And so consequently, for the past couple of years, we've been thinking about how the contents of working memory are actually mapped onto task-relevant actions. So how do you get from a thought to an action? How does that process unfold? And the way that we do this is by using non-invasive tools that we use to measure human brain activity at relatively large spatial scales. So in the past, we've done a lot of work with human functional neuroimaging. The work that I'm going to be talking about today and the work that we've really been interested in for the past few years uses electroencephalography. I know that perhaps not everybody in this meeting is intimately familiar with EEG. So I want to give people just kind of a brief primer about what we can and what we can't do with this particular technique. So in our lab, we can bring in neurotypical kind of standard undergraduate population and bring them into the lab, and we can slap one of these shower caps full of electrodes on their head. And what that allows us to do is to measure synchronous neural activity that's generated by large populations of neurons embedded in superficial layers of the cerebral cortex. So if you've got a bunch of neurons that are arranged parallel to one another, when they're coactive, they produce a dipole that's large enough to actually propagate through the cerebral spinal fluid, the meninges, the skull, and the scalp. And you can pick up on those signals through electrodes that are attached to the scalp surface. And so this is what the raw EEG actually looks like. And like any tool that we have in neuroscience, this has advantages and disadvantages. So let me talk about the downsides first. So the first downside is that it really has lousy spatial resolution. You get tons of volume conduction, so the signal that's generated at this local site can be picked up by scalp electrodes that are several centimeters away. If in a perfect world, if we really push things, the theoretical upper limit on the spatial resolution of EEG is maybe about a centimeter. In reality, it's probably much worse than that. It's probably closer to 10 centimeters. So we have lousy spatial resolution. The other thing is we are limited to activity that's generated by superficial layers of cerebral cortex. Certainly, we're not going to be able to measure anything subcortical. We can't even really measure what's going on in kind of deeper layers of sulci, for example. And another annoyance is that it's just a noisy measurement. So in addition to picking up the actual electroencephalogram, your EEG electrodes are going to pick up ambient electrical noise from the environment. They're going to be influenced by things like skin conductance and a whole bunch of physiological signals that are, in theory, unrelated to underlying brain activity. So it's a noisy measure, right? So, okay, given all these disadvantages, why do we bother with this in the first place? Well, it's got some strengths. The first strength is that it's non-invasive. So we can bring a subject into our lab, put one of these caps on them in about 15 to 20 minutes, and we can record continuous EEG for pretty lengthy periods of time, several hours in a row if we so feel like it. It's fast, so just like invasive electrophysiological recordings, you can get really high tempo precision. So if we push the systems in our lab, I think we can get up to 5 kilohertz. One thing that we really like about EEG is that it is well tolerated by a variety of special populations. So in our lab, we test kind of your typical college undergraduate population, but we have active collaborations with people who do EEG recordings in developmental groups, including infants, in the elderly, and in people with a variety of different psychiatric and neurological disorders. And in all cases, it's pretty trivial to get this data. So it really is kind of although it might be a sledgehammer in terms of spatial precision, it is kind of a Swiss Army knife in terms of allowing you to actually study brain activity in lots of diverse populations. And then the other advantage for us is that it's relatively cheap. So for the cost of a lightly used BMW, you can get a research grade EEG system. So what's nice about this for us is that we can get really high throughput. So most of our studies that publish have 40, 50 participants in them. And like I said, we have these active collaborations with lots of other groups where we're able to look at activity or EEG activity in different populations. Okay, so to give you kind of a brief overview of what I'm going to talk about today, the genesis of this project really began with some work that I did as a graduate student and a postdoc, where we were looking at the role of sensory cortical areas, primarily visual cortex, in the representation of working memory content. And the idea here is that if you need to remember fine visual details about an item that appeared in your environment, you have all this nice perceptual machinery in the back of your head that's evolved to process that information. Why not use that same machinery to help you remember that information? We've done a bunch of studies where we've looked at this and it works, but this poses a problem for us, and that is if visual cortex is involved in representing the contents of working memory, how are those contents ultimately communicated to motor systems that are responsible for producing task-appropriate outputs? So that's the problem that we wanted to work on or that we wanted to look at in the work that I'll be talking about today. And inspired by some of Josh's earlier work and also work by Laura Duguay and Rufin van Rulen, we thought that this traveling wave phenomenon might be a convenient way for getting signals from visual cortex up to motor cortex. And so that's what I'll be talking about. That'll be the bulk of what I'm going to talk about today. As I said at the outset of the talk, this is very early, very preliminary stuff. So really, this is our first foray into this. So I absolutely welcome comments and criticisms and suggestions because like most new endeavors in scientific research, at this moment, we have way more questions than we do answers. So I'll talk a little bit about what we think might be going on and kind of where we're going with this work in the near future. All right. So as I mentioned, way back when I was a graduate student in the late aughts and the early teens, I guess, we were interested in how people remember visual details or how visual details are represented in short-term memory. And around the time, there had been this idea kicking around in the literature that was called the sensory recruitment idea. And it essentially says is that if you have to remember precise visual features like specific orientations or spatial frequencies or hues, you have all this perceptual machinery in your visual cortex that's really fine-tuned for processing that information. So why don't you use that same machinery to help you remember that information? And so in these early studies, I'll just briefly highlight one that we did about 10 years ago. We take people and we put them in an fMRI machine, and we measure activation patterns in visual cortex while subjects are remembering specific orientation values or hue values or spatial frequencies, et cetera. And the upshot of all this is that if you look at the information content of fMRI activation patterns that you get in these early visual cortical areas, you can actually do a pretty good job of decoding or reading out the specific feature value that participants are remembering. So in this plot here, 0 corresponds to the specific orientation value that you have the subject remembering, and then these are other possible orientation values that the subject could be remembering on different trials. So we get this function that peaks nicely. We're able to basically decode or predict the specific orientation value that the subject is remembering. And what's interesting about this is so you can quantify the precision of these relationships or how well you're able to decode the remembered feature value. And it turns out that it tells you a fair amount about how well the person is going to do overall the task. So this is a scatter plot. So the x-axis here is the participant's behavioral error. So in this task, the subject is remembering this orientation value, and they have to adjust a probe to match this specific orientation value at the end of the trial. So the lower the number here, the closer they are to getting the specific orientation value that they were asked to remember. And then on the y-axis, we have our decoding error. So the idea here is in both cases, lower numbers mean better performance. So the subjects who are good at the task overall, who have low behavioral error, also tend to have low decoding error, at least using these activation patterns that we measure in early visual cortical areas. So that's the upshot, is this is support for this sensory recruitment idea, and we have evidence in humans at least for representation of visual details in activation patterns in early visual cortical areas during the delay period of a short-term memory task. We're not the only people to do this, right? So there have been dozens, perhaps hundreds of studies looking at this general phenomenon so far, using both fMRI and EEG. We've been involved in some of this work, some of this later work as well. And we now have, there was just recently a nice preprint that was published by Dake and Curtis now on bioRxiv, suggesting that there's actually a causal link between activation patterns in early visual cortex and memory precision or short-term memory precision, such that if you interfere with activity via TMS pulses, you can actually produce pretty large and pretty specific behavioral deficits in memory performance overall. Okay, so this is an idea, like I said, been floating around in the literature for a while now, and I think we have what I consider to be pretty compelling evidence that supports it at this point. But this has always kind of puzzled us for a little bit, because if these early visual cortical areas are responsible for representing memory content, particularly content for fine visual details, at the end of every single one of our memory tasks, we ask participants to do something with the remembered information. So in the example I just showed you, they get a randomly oriented probe stimulus that they have to rotate and match to the item that they were remembering. We can have people make memory-guided judgments, say, is this probe stimulus rotated clockwise or counterclockwise to the one that you're remembering, so on and so forth. So at some point, these visual details have to be communicated to motor circuits that are responsible for producing the appropriate outputs. And so we've been trying to think for a while about how that process might unfold. And thinking about communication in brains, the first thing that comes to mind is just oscillations in general, right? So there's this classic idea, or now classic idea, by Pascal Fries and colleagues, communication through coherence, where if you want to route information between two different, perhaps distal brain areas, you can synchronize them such that spikes that are generated by one area arrive at a receiving area at some optimal timing or some optimal phase, where they're more likely to depolarize neurons in the receiving area. And there's a variety of different flavors of this communication through coherence idea, but essentially the idea is that you increase synchrony between two brain areas, and that essentially maximizes information transfer between them. So this is a simple example where we have just perhaps two different brain areas, a sending area and a receiving area with some kind of intermediate area in between them. We know from Josh's work and basically everybody who has presented at this meeting for the past few months that this oscillatory activity or this synchrony between different brain areas has a spatial profile to it. So this is from one of Josh's papers from a few years ago, where they were looking at oscillations in intracranial recordings along this axis, kind of running from, I guess, inferior frontal cortex back to inferior parietal cortex. And you can see that there's a pretty clear phase gradient in this one example here, right? So there's a signal that's generated, say, at electrode number 1 that kind of passes to electrode number 2, 3, 4, 5, 6. So a pretty clear phase gradient. So we've known for a long time that there's a spatial profile to these oscillations. And what we wanted to do in our current work was see if we could actually measure a spatial profile that would allow, or a spatial oscillation that would link specific subregions of early visual cortex that we think are involved in representing memory content with specific subregions of motor cortex that we think are responsible for producing task-relevant outputs. And so that was our overarching goal here today, was to see if we could identify a traveling wave that mediates communication between specific sensory and motor sites that are involved in memory storage and response generation specifically. So the way we went about this was we actually went back, way back into the literature and borrowed some ideas from some really early research. So far as I can tell, one of the first demonstrations or the first reports of this traveling wave phenomenon in the brain comes from this paper that's now about 75 years old, This is a quick synopsis. If you read this, it's actually just like a letter to the editor at Science, where they briefly describe a bunch of phenomena that other people have been talking about in this lecture series the past couple of meetings. So this is the authorship list up here, but I want to briefly highlight this paragraph and specifically some of these underlined passages, right? So the most striking activity, however, is that of traveling waves across the skull. In the single lobe, the traveling waves often show a rotational or circular path, successively clockwise and counterclockwise rotations interspersed with straight motions at various angles. So these are all EEG recordings, because this is back in the day when that was pretty much all that was available to physiologists. Now, annoyingly, in this paper, they don't describe how they actually quantified these traveling waves. For that, we went to a paper that was published about 25 years after this early science paper by Paul Nunez, who's kind of a famous EEG biophysicist. And the trick that we used that I'll kind of unpack for you over the next couple of slides is this spatial temporal Fourier analysis of scalp surface potentials from multi-channel recordings. Essentially, you take your electrode sites, you stack them up along some axis of your choosing, and you can apply a 2D Fourier transform to those to get both temporal frequency information and spatial frequency information. And it's that spatial frequency information that define what I am calling traveling waves. We could call them spatially coherent oscillations, whatever you want to call them. But for our purposes, and since this is wave clubs, I'm going to call them traveling waves. All right. So I mentioned earlier that we really wanted to look at relationships between specific sites in visual cortex that we think are associated with working memory storage and specific sites of motor cortex that we think are appropriate for generating task-appropriate responses. Now, we can't really get super specific with scalp EEG because of volume conduction, but we can at least get hemisphere specific. So what we did was we actually borrowed a task from Freek van Ede and other folks in the Netherlands that allowed us to independently manipulate which brain areas should be responsible for storing task-relevant information in memory and which brain areas, or which, I should say, cerebral hemisphere is responsible for producing the task-appropriate output. So here's how it works. So we call this the visual motor memory task. At the beginning of the trial, you're fixating this cross in the center of the screen, and you're shown two tilted bars. These bars flash up for a short interval, and they disappear. About 2 to 2.5 seconds later, the fixation cross changes colors to match one of the two bars that you're being asked to report. And when this happens, you have to press a button on the keyboard. When you press a button, this probe stimulus appears and it starts rotating around in a chosen direction, and you hold the button down until this rotates and matches the specific orientation that you were asked to remember at the beginning of the trial. Now, there's a trick to this, and that is the vertical tilt of the bar that you're being asked to report determines how you're supposed to report it. So in this case, if the bar that you're being asked to report has a counterclockwise tilt from vertical, you have to press and hold a button with your left hand to rotate this probe stimulus counterclockwise, and then again, release the button when this probe stimulus matches the orientation of the bar. If, on the other hand, you're being probed to report the stimulus that's tilted clockwise from vertical, you would press and hold a button with your right hand. And essentially what this allows us to do is to exploit the contralateral mapping of human visual motor systems. So in this particular instance, we have participants being probed to report a bar that appeared in the left visual field. So we imagine that that bar is going to be represented in right visual cortex. And since that bar is tilted counterclockwise from vertical, the subject has to report that bar using their left hand, so engaging their right motor cortex. And so what this allows us to do is, on a trial-by-trial basis, we can vary the position of the bar that's being probed and also the vertical tilt of the bar that's being probed. And so what that allows us to do is make trial-wise predictions about which cerebral hemisphere or which hemisphere of visual cortex should contain the task-relevant information, and which cerebral hemisphere for motor cortex should be responsible for producing the appropriate response. And then we could just basically build a spatial axis across electrodes that link those two sites and look to see if there's any one of these kind of spatial or phase gradients along that particular site here. So we know that this task works. So this is actually, as I said, we borrowed this from earlier work by Freek van Eede's group. And we know that when we present the probe stimulus at the beginning of the trial, we get evidence for selection of visual information and also motor information. So before we did any kind of crazy traveling wave stuff, all we did was say, all right, when we present the probe stimulus, can we find evidence that subjects are covertly shifting attention to select the task relevant bar that they're being asked to report? And indeed, that's exactly what happens. So we get evidence for visual selection. So this is a contralateral. So over contralateral electrode sites, you get a desynchronization or a decrease in alpha power. This has long been associated with covert spatial attention. We also, if we look at kind of frontocentral electrode sites, we get evidence for motor selection vis-a-vis a desynchronization in kind of mu alpha beta power over electrode sites that are contralateral to the response hand that participants are using. So again, left bar, so a decrease in alpha power over right occipital sites. Have to use left hand to recall this. And so we get a decrease in alpha beta synchronization over left central electrode sites, approximately where you would find primary motor cortex. So we know that this task works and that it engages selection mechanisms. So our next question was, can we find a phase gradient or a traveling wave that actually links these occipital and central electrode sites? And so to do that, we use this trick that Paul Munoz described some 50 years ago, where we define an axis that basically starts from occipital electrode sites and propagates up to frontal electrode sites. So here we're going from right so P08 up to F4. These are just kind of canonical EEG electrode position labels. And we can take the signal from each electrode and we can just stack it on top of one another. So time 0 here corresponds to probe onset. And if you just kind of look at the raw data, in some instances, you can see what looks like pretty clear evidence for phase gradients. So for example, here's a peak that emerges at a specific time over occipital site P08. And you can see, looks like that same peak is showing up one electrode up this P6 electrode site, but slightly shifted forward in time. CP4, another electrode site, shifted forward in time again, so on and so forth, right? So even looking at the naked eye, so just like the example from Josh's work that I was showing you earlier, you can identify what looks like these phase gradients. And so the next thing we want to do is essentially just quantify these things, right? So can we quantify these phase gradients? Can we tell how strong they are? Can we tell what direction they're moving in, say from the back of the head to the front of the head, how fast they're moving, so on and so forth. Excuse me. So to do that, what we do is implement the sliding window analysis. So we define a window that's about 500 milliseconds in length, and we just slowly sweep it across the data that we measure on each trial. And within each window, we can build an image out of the activity. So what you're looking at down here, the exact same electrode sites, these are the exact same data, except now we're just looking up in a 2D format, right? So we can take the actual raw voltages from each electrode and just represent them as an image. And now that we've got these in image data, we can extract both temporal frequency information and spatial frequency information using a simple Fourier transform. And so that's what you get over here. And what you get out of this is a couple of pieces of information. So you obviously get spatial frequency on the y-axis, and you get temporal frequencies on the x-axis. And if you subdivide, so these plots are always symmetric along temporal frequency. If you look at the upper quadrant of this 2D Fourier transform, what it tells you is something about negative spatial frequencies. So spatial frequencies that in this anatomical plot up here would have an anterior to posterior distribution. Whereas if you look at the bottom right quadrant of this, it's going to give you positive spatial frequencies. So things that would have anatomically a posterior to anterior distribution. And so this is how we isolated or we quantified phase gradients, or what I'll call traveling waves for simplicity, from the EEG signal. And I should also mention in all the plots that I'll be showing you later on, they're always decibel transformed. So in this case, what we do is we compare the actual data, the empirical data that we get from this to data that we get after we shuffle all these electrode labels. So we break any spatial dependence between the actual recorded data and the electrode labels. So that's what I'll be showing you. So when we do all this, we get a couple of interesting things. So what I'm showing you here, these are plots. So 0 here is time from the probe onset. So this is when you get the go cue that tells you what you have to report, and then you have to figure out how to report it. So immediately after this happens, we get this low frequency anterior to posterior or occipital to frontal waveform that kicks on right after the onset of the probe stimulus and lasts for about a second afterwards. So I'm going to call this the forward wave, and it's roughly equivalent to the theta band, although I don't want to potentially ascribe special significance to specific frequency band labels, but just for convenience, I'm going to refer to it as theta. And then after this theta oscillation kicks off, we get another waveform moving in the opposite direction from anterior electrode sites going back to posterior electrode sites. That's roughly equivalent to the beta frequency. And so just for convenience, what we did was we took all the significant clusters in these plots here and just plotted them independently so we could just look at one time series for these waveforms, for the theta waveform and for the beta waveform. So that's what I'm going to be showing you next. So just to orient here, so this is the power, or essentially the decibel ratio between the intact and shuffled data. Again, time from probe onset. And additionally, what I'm going to be showing you here is this gray window that you see here. This is the participant's response window. So the leading edge of this window is the median response onset time. So when participants first press the button. And then the trailing edge of this window is when the median response offset time. So when they release the button to enter their final responses. So it's response duration. So if we just look at the activation of this forward theta wave, we again see that it kicks on right after the probe appears, and it basically reaches its peak right around the time that participants are beginning their initial response. If we look at the backward beta wave, we see that it has a different temporal profile. It only kicks on after the participant is finished responding and when the theta oscillation is on its way back down. So we thought, this is interesting, right? So we have evidence. So given the kind of temporal distribution of these two signals, we might be indexing two different things. But before I get into what we think these things might be, we wanted to run a couple of quick control analyses that are kind of EEG specific. And I'll just kind of quickly blow through these because I know I'm running short on time. Oops, sorry, I got my slides out of order here. Let me back up. Okay, so our next question was, are these things actually behaviorally relevant, right? So can we actually learn things about participants' behavior? And you would think that that's probably going to be the case since we have one signal that's peaking around response onset and another signal that's peaking after response offset. So to look at this, the first thing that we did was just basically take our forward theta waves and we just broke these down into fast trials and slow trials. So for all subjects, you just take the median split on the initial response time, right? So how long or what period of time elapsed between probe onset and response onset. And when that interval is short, so when there's a fast response time, you get an earlier theta wave that has higher power. When they're slower, you get a later theta wave that has lower power. So whatever we're measuring with this forward theta wave, it seems to be related to inter-individual variability in response times. We can also show that that's true on a between subjects basis. So for every subject, we can just take their average theta wave across all trials, calculate the peak timing of that theta wave, and then plot it against their median response time. So you get this really nice, strong, positive correlation such that the subjects who have earlier theta peaks tend to be faster overall than subjects who have later theta peaks. So that's nice. We can at least say that these things are behaviorally relevant and that they're pretty strong predictors of when participants are going to respond. So next we looked at the feedback beta signal. So in this case, this is a signal that's really only kicking on after the subjects are finished responding. So again, we looked at trials where the participants finish their response quickly, so where they get the bar where they want it relatively quickly, versus trials where it takes them longer to finish their response. And we see the same kind of temporal relationship where when the subjects finish responding quickly, you get an earlier beta waveform compared to when they're finished responding slowly. And there's also a modest relationship between subjects. It's not quite as strong as what we see for the theta wave, but we can at least say that the timing of this waveform that we're measuring here is functionally linked to when participants are finishing their response. Now let me get to the control experiments. So the dumbest explanation that we could think of for this feed-forward theta oscillation is just its feed-forward propagation of the visually evoked potential, right? So we're time-locking our analyses to a change in the color of the fixation point. We know that's going to elicit a phase reset, and that phase reset is going to propagate through even just through passive volume conduction from posterior up to anterior electrode sites. So maybe that's just what we're measuring, at least for the theta wave. And so to test that idea is we basically just took the data. So this is the same data that I've been showing you the past couple of slides with probe locked activity baked into it. So we just took the average VEP across trials, or basically we subtracted out phase locked activity from the data. And we're thinking that might be a good approximation for removing the effects of the visually evoked potential that's consistent over a trial-to-trial basis. And when we do that, nothing really changes. So what that suggests to us is that we're not just measuring an ERP or a VEP at this particular point in time. Something more interesting is going on here. So the next thing that we wanted to look at that you always have to worry about with electroencephalography are eye movements, right? So we know that eye movements can have a pretty profound effect on visually evoked potentials and also EEG signals over frontal electrode sites. So maybe what we're looking at is people are just looking at the stimulus that they're being asked to report, or they're looking towards the hand or the button that they have to press with the appropriate response hand, that kind of thing. And so to test that idea, we again took the exact same data and we sorted it according to, we had concurrent eye tracking, infrared eye tracking data during these recordings. And we just sorted it according to trials where participants moved their eyes towards the probe matching stimulus. So for example, if they shifted their gaze over to the left to the probe that they had to report, or wherever the probe was, or whether they looked to their appropriate response hand, or whether there were no detectable eye movements per se. And the upshot of all this is that nothing happens when you do those comparisons, right? So if you look at trials where there's no eye movements, you get roughly the same waveforms as when you look at trials where there are eye movements towards the probe stimulus or away from the probe stimulus or in any other direction. So again, whatever we're measuring doesn't seem to be a result of just changes in gaze position. All right, so at this point, we're like, what the hell are we measuring here, right? What is actually going on here? So we've got these interesting phenomena, but I can't tell you what they are at this point. And so at this point, we started thinking about, well, maybe they're somehow related to the fact that the person actually has to generate an overt action. So they're given this probe stimulus, they select this bar, they figure out which button they have to press, and that's what's driving this waveform that's connecting, or these waveforms that are connecting the two electrode sets. On the other hand, way back earlier, if you think back to those spectrographs that I showed you earlier, we know that around the same time, even before an action is commenced, participants are selecting the task relevant information that they need. So they're shifting covertly their attention to this bar that they have to report. And maybe there's some analogous process that's taking place in the motor system where they have, for example, two possible motor programs programmed up and ready to go, and they just need to select one of those things. So we were wondering about, okay, maybe we can get some insights into what we're measuring, whether this is a selection process or whether this is a motor process by doing with a couple of additional tricks. So here's the next trick. So what we did in a second experiment was part of it was exactly identical to what we did before. So there's an encoding display where you get two bars to remember, your probe to report one of them. You have to use one hand if the bar is tilted counterclockwise and another hand if the bar is tilted clockwise. So we can do everything that we just did in the previous experiment. But the fun part is that on some trials, we also stick in a pre-cue display. So this is a cue that tells the participant with 100% certainty which bar they're going to have to report before the bars actually appear. So when on these pre-cue trials, it gives away the whole game. As soon as this encoding display comes up, the subject knows exactly what bar they have to report, and they know which hand they have to report it with. And our question was, what happens during this encoding display? If the traveling waves that we measure are all about selection, then if we time lock our analyses to the encoding display here on these pre-cue trials, we should get the exact same thing that we get in our previous experiments when we time locked our analyses to the onset of the probe. So that's the logic here. So during the pre-cue trials, the participants can select and prepare their response as soon as this encoding display comes up, but they can't actually act on that information until this probe appears a couple of seconds later. So essentially what this manipulation allows us to do is to decouple the actual selection of information from the actual implementation of a motor plan based on that information. So we wanted to look at the probe locked activity. That should replicate what we saw in our previous study, but we also wanted to look at the encoding locked activity to see what was going on. So first is just kind of a manipulation check. We wanted to know, all right, when we present the encoding display following a pre-cue, do we get evidence for selection of the appropriate visual motor information? And we do. So these basically the exact same kinds of spectrographs I was showing you in our last experiment, where time lock your analysis to the encoding display, you get evidence for visual selection over occipital electrode sites, and you get evidence for motor selection over kind of central electrode sites. So we know that this particular task evokes selection processes. So the question is, do we see traveling waves around this particular time? So when we time lock our analyses to the probe, just like we did in our previous experiment, we get the exact same thing that we saw in that earlier experiment, right? So we get a feed forward theta wave that kicks on right after probe onset, reaches a maximum when participants begin their response. We get a later backward moving beta wave that kicks on after participants finish their response. So whatever we were measuring in the first experiment, we can show that it generalizes to a different sample. The question is what happens during the encoding display, right? So if this is all about selecting information, visual motor information, then we should get the exact same thing when we time lock our analyses to the encoding display. And we don't. So that doesn't actually hold up. So what I can tell you is that whatever we are measuring with this particular analysis, it is somehow related to the generation of an overt memory-guided action. It is only when you are actually planning and executing an action that you see these particular waveforms, which we think is pretty interesting because it might be a window into understanding how those kinds of processes might unfold. So to kind of briefly orient you to what I've told you so far, and then I'll kind of briefly touch on some ideas about where to go with this stuff next. So I've shown you that you get these directional and frequency band specific traveling waves that link frontal and occipital electrode sites around the time of a working memory-guided behavior. These things are correlated with intra and individual differences in behavioral timing, so we can predict when participants are going to initiate and also terminate their response by looking at the timing of these waveforms. You only get these waveforms when an action is imminent, so they don't seem to be pegged to a selection process. They're really about generating a motor action. And so we think that these are a potential mechanism for the implementation and control of working memory guided behaviors. So I'm sure that many of you have lots of questions, just like I do, of, once again, exactly what the hell are we measuring at this point, right? So we have this general phenomenon that we've now discovered in a couple of independent data sets. It's quite robust. We keep getting the same answers over and over again. The annoying thing right now is I can't tell you what processes or what mechanisms these signals are related to. We have some ideas based on the direction of these signals and also their time. So thinking first about this backward moving beta wave, right? So a high frequency signal that's beginning over frontal sites and filtering back to occipital electrode sites. One possibility, and this is suggested by some intracranial recordings, but in humans, but also some monkey neurophys, is that this beta signal is related to cognitive control. So if you think about what participants are doing after they finish responding, a lot of the times they know that they've got another trial coming up in a second or two. So once they finish responding to a particular piece of information, in order to avoid overrunning their memory capacity, they need to get rid of the information they were just thinking about, right? Either suppress it, remove it from memory, whatever might be going on. So maybe we have an index of that process with these beta signals. Another more germane idea is that, well, participants have just finished responding. And we know that if you look at beta synchrony and EEG recordings around the time of a motor movement, you get this big rebound in beta oscillations when people finish responding. And so maybe that's what we're picking up here. And so one series of experiments that we're running right now is basically trying to adjudicate between these alternatives. The beta waves, we're not really sure what's going on. We think that this might be something related to visual motor planning. So if you think about what participants have to do in these tasks, they know that they're going to start with a vertically oriented probe that they have to adjust in a particular direction to match to a remembered stimulus. Maybe we're measuring something about how long that rotation has to take place. So maybe there's a comparison between the starting orientation of the probe and the actual the orientation that they have to report. So we've got another series of experiments where we're exploring that basic idea. But this is all very early days. I very much welcome suggestions and comments and criticisms about all of this work because this is, we'd like to make this a community effort. And so I hope that given this audience of experts, we'll be able to have kind of a fun discussion about this stuff. All right, I'm over time actually, so I'm going to stop there. Say thank you, NSF, to funding this stuff, Ken Wang for doing all the work. And here is a QR code with a link to the preprint if you're interested. We got a preprint for this on bioRxiv. And I will shut up and take questions and apologize for my dog who's scratching at the door right now.
Thank you, Edward. I don't know if the Q&A feature is working, but if anybody has questions, please post them in the Q&A. Looks like we have a couple questions here. Excuse me. I'm going to pull Nico on stage, or how do you want to do this?
Yep. Can I start?
I see. I've got stuff in the chat. I can just start.
Oh, people have been chatting things to you, huh?
Yeah.
Is the Q&A working? My goal had been to use the Q&A feature. Maybe we're having a hard time. But...
Yeah, the Q&A is working. Let me just put Laura
whatever works. Yeah.
You can ask.
Okay, yeah, go ahead. Yeah.
Okay, well, let me reply to Nicholas, who posted in the chat here. He was asking, have you tried probe stimulus on one side and motor response on the other side to get diagonal waves? So in other words, you've got the visual information. Sorry, let me open my door because my dog's freaking out.
I just want to I can open everybody. Yeah.
Okay. So, yes, we have tried that. So here's an example where, let's say the storage is happening in right visual cortex, but the motor response is generated by left visual cortex. That doesn't work when we do it. And our hypothesis for why that doesn't work is you've got to jump the corpus callosum when that happens. Or you've got to jump the central fissure when that happens, probably through the corpus callosum, which might interfere with our ability to measure phase gradients. We were actually kind of happy when we saw that result because it partially rules out a kind of pure volume conduction explanation for our findings. We just have a signal generated somewhere that's just radiating across the entire cortical surface. So that's my hand wavy explanation.
Wait, but Edward, when you say it doesn't work, do you think that there is a traveling wave that goes across the two hemispheres that scalp EEG is just not measuring? I just
can't get it, right? Because so one of the challenges that we face here is I don't know what is generating these waveforms, whether it's horizontal connections or whether it's steeper connections. And so, as I said, our hand wavy explanation is that to get across the central fissure, you've got to go through one of the commissures or the corpus callosum, and that might induce an extra phase delay or phase lag that interferes with our ability to detect these waveforms through this kind of spatial frequency-based analysis. We could probably figure that out with some simulations. We just haven't gotten that far yet. But when we try to do that specific analysis, if we just look at the waveforms over time, you just get a bunch of flat lines. You don't see any evidence for peaks or any directionality or anything like that. So I do not have a detailed explanation for what might be going on there. But in some sense, as I said, we were somewhat comforted because it rules out pure volume conduction, where we've just got a signal generated at one point under one electrode that's just radiating everywhere across the scalp.
Great, thanks.
Yep. Laura has questions.
So you can actually unmute and turn your video on now. Yeah.
Yeah, we decided to
Hi,
take all the
sorry.
restrictions away. Everyone can turn their video and camera on audio on if they would like. Like Laura, if you would like to, go ahead. Yeah.
Hi, thank you. Eddie, thank you. I have a bunch of questions, but one was already asked by Nicholas. I actually want to go further on that because I'm not sure that the 2D Fourier transform is the best way to address Nicholas' question. So maybe there are multiple ways, and that would be actually really cool to imagine like one way to address that volume conduction problem would be speed, because if it goes through the corpus callosum, it should go faster, right? So ultimately, if we have a better way to measure the diagonal and you see a speed difference, that would rule out volume conduction. But anyway, yeah, we can discuss this further. I don't want to re-ask the same question. I have another one. I was very happy about the link between the wave and the reaction time. That was actually really, really cool because it means somehow they mean something, you know. Right. That's kind of reassuring. But I was surprised that you looked at reaction time actually, because with the task that you have so much more you can do. And I was thinking in particular about the error in orientation report. So I was wondering if chance you looked at other feature bonds that could be diagnostic. Okay, you have slides. That's cool.
So this is just so that was our first thinking, right? Because the real important variable in this task is the recall error. That's what tells you how well you're doing. I mean, we do tell participants respond as quickly as you can, but also be as accurate or as precise as you possibly can. And so in this instance, when we just took the participants' trials and we medium split them into high error trials and to low error trials, we didn't see any difference in the waveform timings or the waveform amplitudes, nor did we see a relationship on a between subjects basis. Now, I will say my suspicion is that some of this may be caused by essentially a ceiling effect in the task. So the majority of our participants, they're already responding with an average error of 8, 9, 10 degrees, which I can tell you from personal experience is pretty much as good as people are going to get for being able to remember the bar of a stimulus that's 5 or 10 degrees in the periphery of their vision. So I suspect that we just don't have enough variance to be able to detect links between these waveforms and overall memory performance. And so that's one thing that we're actually pursuing right now is we've come up with analogs of this task where we can make it harder by having people remember more stuff to get us a little bit more variability to see if there's actually a relationship between these things.
Yeah, that's very cool. I think you can really have a diagnostic tool for it with the task. It's a beautiful task. Yeah. And I think actually the reaction time, the problem, if there's one, with reaction time is that it's very much subject to decision biases. So when you are at the stage where you try and disentangle what it is about the task, right, that is correlated with my traveling wave, you really don't want this variable to bias your interpretation. So I think you really have I really like the task. I think that's the cool way to go about it. And we can discuss further the analysis together if you want.
Yeah, we've been thinking, so in some ways the analysis that we're doing is very simplistic because it's only allowing us to measure activity that's along an experimenter defined axis, right? And we can only look at what's happening going anterior to posterior, posterior to anterior. We know that there's way more kind of interesting dynamical stuff that's probably going on underneath the hood, right? I mean, that science paper from 1949 was reporting rotational waves
Yeah.
in scalp EEG. And so we've been strategizing about tools where we can really dig into these data and look at perhaps maybe higher order dynamics that might be more precisely linked to different aspects of behavior in this instance, and might be happening before versus after responses and things like that. So we just haven't had enough time to really sink into it because poor Ken Wang's got to set up his lab.
Happy to share the tools that we have.
Oh, that would be awesome. Yeah, I would love to have a chat about that. By the way, I know I owe you an email, so I'll be in touch soon.
That's all right. Okay. Thank you.
Lee Howe, I see you have a question. I don't know if you want to unmute yourself and ask.
Yes, thanks. Thanks for your very interesting talk. So I just want to know, how do we interpret the traveling wave's direction in terms of the information flow? Because for a wave, you can think about it as one end pulling across the rope, so it's traveling wave in this direction, or it could be the other end sending out the wave. So I could think information flow in both directions when I observe traveling wave in one direction. So how do you think about this?
That is an excellent question and something that's we'd really have to dig into the biophysics of EEG to get into that. So the short answer to your question is I'm not sure. The way I can tell you that we've been kind of conceptualizing these is, look, we're picking up on local dipoles with our EEG electrodes, and we're just looking at phase relationships between them. Another possibility is that there is some larger scale, almost kind of, I don't know how to describe this, larger scale kind of series of sources and sinks where you get this kind of gravitational pull towards a particular location, and that's what causes what we're calling forward or backward propagation. I suspect we might be able to tackle that, or at least examine that possibility using some of these other kind of more advanced analytic tools that Laura was just mentioning, where we could look at kind of higher order dynamics. And also it would be nice if we had higher density EEG recordings. Right now we're limited to just a small number of electrodes, about 30 per hemisphere. So the short answer to your question is I'm not sure. But it's one thing that we're thinking very carefully about. So, one major limitation to all the stuff that I presented today is we just don't know what the underlying biophysics and the mechanisms are. And that makes it really difficult for us to draw conclusions about what specific processes or what specific cognitive mechanisms that we might actually be measuring. And so that's something that we were really hoping to kind of sink our teeth into going forward. So I thank you for the question, but unfortunately, I don't have a good answer to it at this point in time.
Yeah, thank you.
Hey, Edward.
Yes.
Pascal Fries had a paper about 9 years ago. I don't know if you're talking about how theta, this is a very different task, of course, but talking about how theta oscillations were feed forward, beta
Really?
oscillations were feed back. He didn't exactly, it was Andre Bastos in Neuron 2015. They didn't exactly look at it in terms of traveling waves, but I think it's totally consistent with what you see. I was just looking at it
No,
here in case you heard me typing because I forgot to mute myself.
no,
And
no. Yeah, that's I'm not familiar with that paper, but I'm going to go look it up as soon as we're done here.
I'll send you a link. I think it's totally consistent with everything you've seen.
All right, awesome. No,
So
I would
anyway,
love
that might answer the
to.
question that Lee Howe had. I don't know. Feed
Yeah,
forward.
yeah.
Feedback, yeah.
I had a question building on sort of related to Lee Howe's, and that was I guess you don't ever go as far as the frontal cortex in looking at any of these waves also, do you at all?
Well, we get close. So let me find my little scalp map here. So this is, yeah, F4. So here we're probably over kind of posterior lateral frontal cortex. But Yeah, we haven't gone all the way up to, so problems you get with AF4 and FP2 is now you've got people furrowing their brows a lot. And if you've got any lingering eye movement artifacts, that's where they're going to be the biggest. And those are definitely going to radiate from, for example, anterior to posterior. So this is kind of a happy compromise for us.
I was trying to connect this a little bit to the stuff with Uma and Josh on the directionality and whether
Yeah.
you're coding or and whether in this case, I guess the decoding here is more the motor. I mean, is the motor action is more would be the analog of that of the playback. So I guess, and that wave, what direction did that goes from
That
occipital?
goes from anterior, the beta stuff goes from anterior to posterior.
Okay. Wait, but the beta the motor wave goes the other direction. Okay, from not
Yeah.
it goes from motor to visual.
To occipital, yep.
Yeah. Huh.
Yeah, so whatever, I mean, so that's why we thought this was really interesting is we've got two signals going in two different directions at two different times in two different frequency bands. And so what we're trying to do right now is just make sense out of what the heck is this? Like I said, I'm more than open to suggestions, but it sounds like this paper with Andre might have some hints in it. And yeah, we've got a ton of work to do on this stuff.
I had a question about, so one of the things that I've always found interesting and mysterious about traveling waves in my lab's work, which is based on intracranial EEG, which is different than this, of course, is that we really see differences across people. And I wondered, and you had at least one plot that showed that you were measuring the latency of waves in individual people. And I was just wondering if you could say anything about how consistent the effect, yeah, that plot,
Yeah.
the effects are across people or not. Yeah.
So they're pretty, we get a fair amount of variability in the timing of these waveforms across people. So, I mean, we've got our fastest folks here, we're getting peak latencies about 400 milliseconds after probe onset, and our slowest folks, they're close to 2 seconds after probe onset. And it's not a perfect linear correlation with median response time, so the slope of this isn't equal to 1. But we do see on an individual subject basis that there's quite a bit of heterogeneity, at least in the timing of these things. Now, in terms of their amplitude, it actually seems to be pretty low. There's not a lot of amplitude variability overall in that, so these are actually, I think these are SEM, these shaded regions. So the 95% CIs are going to be a little bit bigger, but I believe this is 25 participants. So this is not, a ton of variability, especially for a signal that's as noisy as EEG. So I can tell you that there's a lot of variability.
And then also you showed the single trial traces, which were
Yeah.
nice. And I guess I'm kind of wondering, how often do you see traces that look that nice?
Well, that's a good question. So we definitely cherry picked this result, right? Just for a nice demonstration here. I would have to ask Ken Wang because he has spent far more time looking at the raw data than I have to see how consistent these things actually are. Because I know that, in your work, you get these beautiful phase gradients.
No, but we don't see it all the time also.
Yeah.
I think one of the mysteries to me is if these phenomena, like you showed really nice data to show that these correlate across trials on average, but they're not there every trial.
Yeah.
And that's interesting to me.
Sure. I absolutely agree. Because obviously on those trials, they're still doing the task. Maybe they're just faster, they're not optimal at it, but they can still do it. And so what's going on there?
And then the other question I had, you had I think it's 2 slides forward. You had it on the screen before, the scatter plot. It looks like people are responding before the wave has its peak.
Yeah. No, so sorry. Let me
the scatter plot shows that, right?
Some people, I guess like a few.
Some people, although these are this window here. Yeah, so I mean like the median response onset time is about 700 milliseconds.
Okay.
And so yeah, you are getting I mean, I would have to go back and look on an individual. Well, actually, so you've got peak forward and latency about 500, and median response start is about 450, 500. So they're pretty closely linked, but there is some variability around that. It's not a perfect relationship.
Okay.
So yeah, there are lots of potentially interesting aspects of these data that we just haven't plumbed yet.
Very cool. Thank you. Are there any other questions? I
Well,
think,
All right, should we wrap up, Ooms?
yeah.
okay. Well, thanks everybody. I really appreciate the audience and the feedback. And again, if you're interested in this work, there's a preprint on bioRxiv, and we're going to have lots of fun playing with this stuff over the next couple of weeks. And again, thanks to Josh Bard, Uma, and Irfan for organizing, and I'll look forward to the next meeting.
Edward, this was great. Thank you so much. I checked out the preprint while you were talking, and it does a nice job explaining many of the things you're talking about, which is great.
Absolutely, yeah, no, great. So, yeah, if anybody, if you have any lingering questions or anything like that, feel free to shoot me an email. But yeah, again, I'll look forward to the next meeting and see you guys all on the conference circuit.
Sounds good. All right, take care.
Yep, take care.